Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is February 19th, 2026, and we have Dr. Dayu Lin with us. Dayu is Professor of Psychiatry and Neuroscience at the Institute of Translational Neuroscience at Langone Medical Center at New York University.
Yes.
And her lab studies brain mechanisms of innate social behaviors, including mating, fighting, predation, and parenting, all of which are pretty complicated. And these are not simple reflexes. They're also not complicated cognitive behavior that requires the whole brain to be engaged. And so we kind of expect that there will be particular places in the brain that are active and they're generating these behaviors and that underlie them, and that we could discover those and not have to understand the entire brain in order to make progress.
Yes.
And so that's what Dayu does. She tracks down the circuits using behavioral, neurophysiological, neuropharmacological, and genetic approaches, and she dissects the individual contributions of these circuits. I hope that's okay. Welcome.
Thank you. Thanks for having me.
Also with us, Tony Burgos, podcast favorite local social behavior expert. Hi, Tony.
Hi there.
And Aayushma Kunwar, a PhD student in our program, who also studies social behavior. Is this the first time you've been on the podcast?
This is my debut, yes.
Welcome
Hello,
to
everyone.
Neuroscientists Talk Shop.
Thank you. Happy to be here.
I'm your host, Charlie Wilson. So, in a simple way of thinking about it, which I do, the simple way of thinking about it, any kind of complicated behavioral sequence, it's not just a reflex like the iris reflex or something, requires both brain circuits that can compose the movement, that can create the sequence of motor neuron activations that are required to make that movement, and can be flexible with changes that happen during the course of the movement. Yep. But we use the same set of motor neurons and kind of the same set of neurons all the way through the movement, like it's just the temporal sequence of them, and this motor neuron fires less and more during the movement, but it's the same motor neuron the whole way through.
Yep.
And so we could kind of think, well, our motor system, it's very general purpose. No matter what I'm doing, I use the same motor system. But we all, in addition to that, we need something that chooses what we're going to do.
That's right.
And the thing that chooses what we're going to do, it seems like it's usually not like a general purpose thing like that. Like there's a part of the brain that's active when we choose this, and another part that's active when we choose that. So is that true? And if it is, can you tell us something about what circuits we should be looking for with social behavior?
Yeah, my pleasure. Yes, absolutely. This is very much true. The parts of the brain that controls those innate social behaviors are highly conserved and being recognized that likely being innate. There's a parts of the brain and across the vertebrate systems are important for these survival behaviors we consider it. And because they're so critical, it was believed that they're probably hardwired circuit in some way. That hardwired means developmentally wired. They are not required for experience, for learnings, for those circuits to be existing, be activated. In fact, when the animals has those circuits, when there is the right circumstance, when they find a mate or they find a young, when the young appears, they kind of know exactly what to do. So it has been, approximately a century, I would say, people trying to identify where those regions are. And hypothalamus become a key region that was recognized very early on. It is an important part. So in the very early days, the experiment was involving, when we think back now, was really kind of a course in a way. So in the experiment, it was largely cutting the brain to half and see like what's happening if you cut it to half. That was mostly done in the cats. And it was finding that if you cut the brain to half, and in some cats, when they wake up, they become spontaneously very aggressive. And in other animals, they don't. So, and it turned out that the animals become spontaneous, very aggressive. The cut was made anterior to the hypothalamus. And that was disconnect the anterior part, the cortical, any control regions, and now we know the control region, we consider them, detach them from the hypothalamus. And then if you make the cut posterior to the hypothalamus, then the animal don't show that.
So presumably in that case, the animal's still aggressive, but only in its heart, because it can't actually move in relation to aggression.
They actually can move. They just don't show it. So that
so the anger or whatever is disconnected.
Is gone. So then that comes the idea that, okay, so hypothalamus is this critical area that you need to generate the behavior. And it is under the tonic control of the structures anterior to the hypothalamus that suppressing the animals from expressing it.
So we would be angry all the time if we...
Yeah, I mean, not knowing more about it, perhaps it's actually do seem to make some sense. I'll tell you more as the hypothalamic region we study, which does seem to be under a lot of tonic inhibitions. So that's probably is a good thing. But the hypothalamus has become kind of this kind of a region people start to zoom in saying, so this seems to be the important place, those innate behaviors likely to reside. And then that comes, moving forward when people start to use electrical stimulation. That was in the 1940s, still in the cats, and showing that if you stimulate different parts of the hypothalamus, then the animal will show different kind of behaviors. So some parts you stimulate, the cats will show hissing behaviors, and some parts you stimulate, the cat will go kind of attack a rat. So there's a cold blood attacking predation behaviors. So this really solidified the idea that the hypothalamus is this important areas. So, and after that, I have to say, research is a repeated search. Yeah. So we really now down in the cats, and then later on in the 80s, basically a lot of detailed mapping down in the rats, and still electrical stimulations and trying to map out different parts of the hypothalamus. What do they do? You find all sorts of stuff there. It's very interesting. Yeah. Some parts you activate again, showing freezing fear. Some will show teeth chattering. Some will show grooming. Some will show attacking. So there's this map out there that we kind of know this part is the hot area for this and that's for that. So now, but there is not a clear anatomical correlation very clearly, partly because the electrical currents, they just kind of spread, right? So it's very hard to localize very precisely. But you can get the idea that the hypothalamus probably contains many cells that drive different parts of these innate behaviors, social and non-social. And then that goes down to from the rats. In the last 20 years, we kind of reinvented the field in the mice. So with the help of the optogenetics, which allows to target much more specifically different parts of the hypothalamus. So that comes to our research, which being part of this new trend. And we identify different parts of the hypothalamus able to driving very realistic, naturalistic, innate behaviors. So one of the regions we identified is called the ventrolateral part of the ventral medial hypothalamus. It's a small subnuclei in the hypothalamus. I should say that hypothalamus, it's different from the cortex, which has this beautiful layer structures. Hypothalamus is kind of messy. So I consider it's kind of an island in the ocean type of organization. So there's a concentrated island, which is a nuclei. And then there are just cells that scattered all around it. So VMH is one of very important nuclei in this one of the island. And then it itself is complicated because it can be subdivided into probably 5-6 different components.
And how do you do that? Because if you just look in the microscope, you're not going to see all these components usually, right?
Yeah, so that's where really the genetics has been very helpful. So in the early days, when we are doing the optogenetics, it's very much the wild type animals, right? So we're using the wild type expressing channel rhodopsin and just hoping that we're hitting that spot. And I can tell you the success rate is not very high. You know, among the 10 animals we do, maybe 3 able to show it, right? And then over the time, we start to figure out, oh, there is this cluster of cells there in the right locations we really want to target, and they express this particular gene. So, and then now there are a lot of Cre lines available, which allows us to target those genes. The genes mark the cells. So that's really what has been changing the field so much, which as it allows it, even in this kind of a salt and pepper mixed populations, you're able to isolate a particular subpopulations of interest. So, but I do think hypothalamus, you know, again, different from the cortex, the cells there, even though they are having some different roles, but they do largely topographically organized. So in different subcompartments of it, they do have a relatively homogeneous role there. So when we activate those populations, what I consider the behavior, this output is telling me the animal is actually able to interpret these signals in the hypothalamus when those cells become high. That is different again from the cortex, which you just activate you blast the cortex. The animal will be confused. They just don't know what's going on there. But the hypothalamus, because those cells, they kind of drive one singular behaviors. When the activity there is high, the animal is able to know what to do, what those high actually means.
So you can see these cluster of functionally related cells because they express a common marker.
That's
of some
right.
kind.
That's right.
So,
yes.
and how many? So using this method, how many different clusters do we think there are, and how many different markers do we have?
Yeah, that's a very good question. You know, we now know, even within the hypothalamus, even within this tiny region, the VMH, right? We found that the VMH DM, the dorsal median regions, which express this marker called SF1. It's not exclusive, but it's enriched there. And at that part, we consider when we activated the animal show very real fear-like and freezing, flight, escape, jump behaviors. So this is like a defense area. And just neighboring that, which is the ventrolateral compartment, when we activate it using another marker called ESR1, then the animal show attack. So, and if we move this is what I'm talking about is the posterior, the bigger part. But if we move it to anterior, the VMH gets a little bit smaller. But there, when we activate the ventrolateral part, you actually show this is social defense behaviors. So this is the part the animal use to actually avoid conspecifics when they got defeated. And in the females, it's getting also complicated because in the females, this posterior VMH-VL part contains 2 subdivisions, and one part marked by the NPY2R, another gene important for aggression, and then another part in the lateral part marked by CCKAR, another gene important for female sexual receptivity. So, you know,
So I'm not sure, but I've been counting on my fingers, and I count something like 6 different functionally related clusters within just the ventromedial hypothalamus, and that's only one piece of the hypothalamus.
That's absolutely right. Yeah. So
perhaps the diversity of all these hormones or specific receptors is giving nowadays advantage given all the genetic tools, right?
Yes.
And what seemed to be a disadvantage for hypothalamus researchers nowadays is golden, because the analogy I was making earlier today during your talk, you know, like activating or inhibiting these clusters, they serve like light switches, right? You flip them on and you obtain very strong behavioral outputs, right?
That's right.
Yeah.
That's right. I really think, the research in the hypothalamus is tremendously benefited by these new tools. The optogenetic combined with the recent tools for recording genetically defined populations. Now, even though they kind of all mixed in together, but you can have ways to tease them apart.
Yeah.
And because of those cells, once you tease them apart, the subpopulation plays a relatively homogeneous role. The animal can interpret. It can drive a clear behavior output. So now you can link these things together, the response of the cells, the activity of the cells, and the behavior output. And now you can understand, okay, so if things change, what's going on? So you have a very good neuronal handles of the behaviors in those circuits. That's what makes it so much powerful now.
So do you think within one of these clusters, the cells are all interchangeable? They're all got exactly the same function. They're just, we've got, I don't know how many of them, maybe not a huge number, but we've got all of this kind of in the form of cellular redundancy to make sure that the thing works.
Yeah, I do. That's a big conclusion to draw, because in a way, cell clusters are, you can kind of make the cluster smaller and smaller, right? If you do based on the transcriptomics, there's always different ways you can tease them apart. But I do think, yes, there is we don't know whether we have found all of them, but I think for some of them, likely. For example, the CCKR, which is the population for female sexual receptivities. And when we look at this population, this is like the most beautiful overlap I've seen between a gene and the immediate to early gene CFOS expression. They have like 70-80% of the overlap on each direction. So, which really tells me that almost all the cells that involved in these behaviors are expressing this gene. So this is very rare examples that we can see the functional and the genetic markers has lined up so well.
Those are very high numbers, right?
It is a very high number.
Compared to other parts of the brain and even amygdala regions that we typically, kind of don't talk about the hypothalamus when we talk about threat and avoidance and all these adaptive behaviors, but we never see anything like these numbers, right? But it also brings me to the point in which, for example, how many roles, these islands of cells, expressing certain genes, how many functions do they have? Do they only have the function that you're investigating? Because they seem to be like good targets for trying to provide some kind of treatment, for example, like reducing aggression. Well, perhaps do some kind of ablation of this specific cell population. Is that true? Do you see other impairments if you remove those neurons?
So if we inhibit this very specific population of aggressions, at least, other than aggression, it's hard for us to tell what is anything wrong with these animals. They seem to be socially intact just fine. These cells, they are normally very quiet. If you have an animal not engaging those situations, if you look at the immediate early gene, which is a way to assessing the neuronal activity, they generally are very blank. This area, they are not activated. So they are kind of reserved very specifically for these particular situations to make sure that the animal don't get confused when they need to do that. But that said, VMH actually has another very well-known role. And the people study that role, which is satiety, metabolisms, are very much separated from people who are studying social behaviors, as if we're talking about completely unrelated structure. But the cells, in fact, they're exactly the same cells. So now the question is, why those cells, they are able to do both? Are they sensing those metabolic signals, all the cells, they are glucose sensings. So are these cells sensing those signals to tell the animal whether it is a good idea to attack or not? Because, if you are in energy deficit, maybe you don't want to waste your energy to attack another animal.
Or maybe you're hangry.
Maybe you are hungry, so you should really go for it so that you can compete for the food. So yeah.
I think it makes sense, in terms of, for example, anterior cingulate cortex, right? Like we talk about pain perception, right? A lot of encoding through anterior cingulate cortex. But if you think about, what aversive learning is some kind of emotional pain, if you will, right? Emotional from physical pain still being processed or engaging many subsets of cells that are very similar to each other. Perhaps, not so different from what you're talking about, that satiety could be this kind of, or inducing this emotional state that requires, like this kind of being hungry or some kind of madness. And I think it gets into a little bit in the question I asked earlier, which is like, all these behaviors are very complex behaviors, right? Like, at least from the videos you showed, the examples that when you do like a stimulation of these subsets of neurons, then you get inducing aggressive behavior or parenting, right? But those look very purposeful, very organized behaviors, right? Even when in your stimulation experiments, for example, all the cells are getting stimulated at the very same rate, right? So how do you explain this? Is there a driving of the behavior itself, or is it driving like a feeling that is producing that kind of behavior?
Yeah, that's a very good question. Actually, it has been a forever question. Ever since in the early days, even with the electrical stimulations that you can release those behaviors, there has been this debate, is that basically activating a stereotype, the sequence. It's a sequence generator, stereotype, the action pattern generator. It is actually inducing some sort of motivation and emotional state, put the animal in, and those behaviors are kind of a born out of it, right? So I think this debate, to this point, at least in my view, I think that there is overwhelming evidence actually supporting probably it's the state. It's a state change rather than the sequence generators. So it's not, the hypothalamus is not a premotor area, it's not a motor cortex by all means. It does eventually talk to the premotor areas in the midbrain. It very directly influencing on the premotor neurons, how they are going to react.
Premotor neurons where?
In the midbrain, in the periaqueductory brain.
Okay.
Yes. So that's actually the major projections on the VMH goes, and almost all the hypothalamus cells actually. But they themselves are not coding any moment-to-moment movement. So in our ePhys recordings, we're never seeing the cells got activated only during this particular actions, during a particular bite. You cannot decode those kind of informations from those cell activities. So those cells are also very much sensitive to, in the mice, to the olfactory cues from another animals, which is really the most important sensory information this animal need to use to identify the other animals. What is the identity? Because in the males, they're pretty gentle mice. They're not gentlemen, they're gentle mice. So they don't attack females. Male mice very much exclusively attack male mice. So these are the aggression-provoking cues. And for the mice, it's important that they identify the target is appropriate. You don't want to attack a female when it comes to your homeland. You want to, get rid of other competitors. So it's critical for those cells to be able to know that this is male intruders.
Mm-hmm.
So the cells are very tuned to them, right? And those cells, what else do they respond to? Because of their glucose sensing, and they also has all these sex hormone receptors. They have, I mentioned NPY2R, so which probably allow them to sensing the hunger state of the animals. So they're sensing a lot of internal state the animal is at. And from our studies, we also find that those cells, the property of those cells, they really change with the experience a lot. For example, in the animals that repeat a winged fight, then those VMHVL cells, their excitability just goes up. So now, basically, the state is kind of encoding the whole history of these animals at that moment of time, the physiological state of this animal, as well as whether the target is appropriate or not. So this information combined to determine how active these cells are. And that is, they're signaling is aggression an appropriate behavior output. Yeah. When they are high, they say, yes, you're at this good state, physiological state. You have winged many times, so you probably have a good probability of winning again. And this is the target, the right target you should fight for. So now all these informations are being sent to the midbrain structures. And we consider the midbrain structure, I consider them probably plays a very important gating role, but not necessarily be able to drive those cells right away by themselves.
Right. Mm-hmm.
There need to be some additional input because the animals, when they are having all these high signals, but there is no target, right? We can drive those cells to fire very high if we just let them to smell the other animals' urine, for example. They smell the urine, the animal is very high, but there is nothing there for them to target. And they're not going to go crazy and they're just biting everything they encounter. Mm. So, but when there is this other mouse there, and now this immediate sensory input, which I think, has certain features, but probably very loose features, such as that it has some soft skins, textures that they can bite on, and then they will unleash these attack actions onto those animals. So that's kind of how I see it. It creates a motivational state which enable the premotor neurons to react to these immediate sensory cues when they encounter it.
And so all these internal states, right, and motivations, they go back millennia, right? They have been evolving, right, through evolution, and we don't like talking about, like feelings in animals, right? Which in my opinion, is kind of silly, right?
Yeah.
I mean, all these primordial functions, right, have evolved into more complex things, right, and feelings in humans. But to me, they sound like the basis of what we know as complex emotions nowadays. What do you think, Aayushma?
I think motivational states. So when you were talking about motivational states, I was thinking about how if amygdala is a region for kind of marking positive valence versus negative valence, and how hypothalamus could, in addition, be then marking social importance or not social importance, and if that's the kind of motivational state it provides.
Yeah, absolutely. I think it provides, you can consider it a motivational state, you can consider it as a need state. Mm-hmm. So the hypothalamus not only has all these social related regions, but also regions related to hungers, to thirst, right? So to even thermal needs. So there is a lot of need state signals. And actually going back to what Tony is saying is about the feelings. So feeling and emotion or motivation, these are, all big words.
Yeah.
But there are plenty of animals, vertebrate species, that they don't have a cortex. And they have all these social behaviors, innate behaviors. they probably have motivational state.
Yeah.
Do they have feelings? I'm not sure, because I consider feeling as an subjective interpretation of those motivational state, and there we are consciously aware of them.
Right.
So that's when, the difference between a state, a motivational state, to become feelings.
I see.
If they don't have the cortex, they probably don't have the substrates needed to be consciously aware. So do they consciously aware that they are in that state?
This is very similar to William James, the James Lang theory, that you feel hungry because you
interpret
detect all of the signals of hunger.
Yes, I absolutely. So that's like the bottom up inference is a huge.
Right.
Yeah, we didn't do it for ourselves, that for aggression, but it has been done for hunger state, right? So if you activate parts of the hypothalamus, which really controls hunger, that drives the animal to really seek for the food, those cells that they are detecting generally the physiological state, those peptides that get released when you're hungry. So those things, when you activate it, you see the whole cortex, how they respond to the food, become very different.
Right.
So it is really have this bottom up a huge inference, and the likely hypothalamus, other parts of the hypothalamus, when we are have this high aggressive state, how the cortex will respond to the social target will be different from when you have these other regions which control the sexual motivations being activated, right? So,
if I may, correct me if I'm wrong, putting it together with what Aayushma was talking about. So we typically in our field talk about emotional valence, right, for external cues, right? But perhaps the hypothalamus could be seen as some kind of valence provider or reader based on internal signals. Is that correct?
Well, I think hypothalamus, I consider the state created by hypothalamus is a bit more specific than just a positive or negative valence. And I do think they tap into the valence systems very robustly, which, you can maybe consider the dopamine system is in the ventral tegmental areas, being largely encoding the valence signals. The valence in many parts of the hypothalamus, if you activate those cells or inhibit those cells, you can see that they are having a valence, because in this type of a task, if you allow the animals to choose whether they want to be self-activated or not, some regions will very robustly, they want to be stimulated. In other regions, they want to be not stimulated. So that tells us that there is an intrinsic valence for all of them. And for many of the hypothalamus regions, we now know actually you can change those when you activate or inactivate those regions, mostly done in the activation forms, then the striatum dopamine systems will change. So they have ways to tap into it. And we think that that's important because hunger, for example, is very negative valence. And then there are other things which is such as copulations, sexual behaviors. They're very positively valenced.
And the other topic you were talking today about parenthood, right?
About parenthood, they are also causing dopamine release. So all these things, I think, but it's important to, however, in my view, I don't think the valence is the reason the animal doing those behaviors. So they are associated with the experience of those behaviors, which then help with associative learning, not to reinforce that behavior itself necessarily, but the context, the subject that associated with that experience. So next time they will be able to learn it more effectively. So like winning a fight, then they start to have established this preference for this area that win the fight. So that is related to the valence associated with this winning of this fight.
Right.
So Yeah, so these are two things importantly, because actually we find that dopamine plays very little roles to reinforce aggression itself. Yeah. So even though dopamine is released in the accumbens when they win the fight, but if you block this release, the aggression perfectly fine, and they will still become more aggressive as they win more fight. So the aggression itself does not depend on that. Wow. It's the context and the other things. Yeah.
We've been talking a lot about fighting, but some of the stuff you showed was this very gentle parenting behavior that mice do, and all sort of controlled in the same way by nearby, sometimes even overlapping regions in the hypothalamus.
Yes, yes.
I don't know. I just wanted to point out that it is all fighting.
Yeah, that's right. So so the hypothalamus has been, there are some very well-known major nuclei that has been assigned important roles, right? So the VMH, for example, is important for aggression. That's probably now its most known role in the social behavior context. And another region, which is anterior to the VMH, called the median preoptic nuclei, also a big area. They actually really talk to each other very closely. But that area has been known for decades, important for male sexual behavior and parental behaviors. So, but what's kind of funny about the MPOA and the VMH-VL is that they seem to really operate in very different ways. So for one, the MPOA cells, that's important for the parental behaviors, all male sexual behaviors appear to be largely inhibitory. Versus in the VMH, they are largely glutamatergic cells. So which kind of, glutamatergic are more straightforward. You activate it, downstream activate it. The inhibitory cells are much more complicated, exactly how they're going to influence the downstream cells. Why inhibiting the downstream cells will cause those behaviors to be released.
Right.
Yeah, we don't have time, I guess, to go into it, but if we did, what I would like to talk about is the actual circuitry, because it sounds like those neurons are part of slightly different circuits, and these neurons, none of them live kind of in a world of their own. They receive input from lots of different places. That what they do depends on that, and then they send their output specifically to other places.
That's right.
So this is still something we don't know everything about, and I think the anatomical vagueness of the hypothalamus really held back work on that kind of thing, and also the fact that many of the connections they don't form discrete pathways, big strong pathways that go to one other place. They're largely interconnected with each other, and then they have pathways that go to lots of other places. And we can't really tell what's the most important one of those.
That's right. That's right. Yeah, they are very interconnected, and aggression in particular, actually, to this point, it has got quite a bit of attention, and many labs has looked at the aggression in many different places. There are at least 10 different areas that people are able to activate it and get aggression and inhibit it and suppress aggression. So what does that mean? So in one of our recent studies, we recorded certain brain regions simultaneously in the limbic systems. And if you look at aggression, virtually all of them will get activated. It's hard to say who is the first one to get activated, but they are synchronously activated. So I think the only way to explain that is it's a very heavily interconnected network. So you can perturb the flow in this network by blocking probably many of the nodes. And you can trigger this network by activating many of the nodes. That's why the aggression is seemingly so easy to be activated from many different places, as well as being inhibited from many different places. And talking about the parental behaviors, I think it's a very different way to organize it. Probably because there's so much inhibition there. In fact, I think MPOA is the only place we know that really can perturb it and can activate those behaviors. We still don't know the upstream, still don't know the downstream. So I think it's different principles of how the circuit is organized.
We got 9 places for aggression and just 1 for kindness. And that explains a lot of stuff.
Exactly. Yeah, it's not the lack of effort. There's actually plenty effort going there. Yeah.
It's a mix of very complicated, functions of the brain that, in my opinion, is going to be really hard to replicate for a robot, for example. If you were to try to introduce these functions in a robot, but perhaps robots are just based, or at least for now, are going to be based on motor commands or motor functions. But it seems that we are talking about here about motor functions or complex behaviors, right? Which require motor functions, but they are being modulated or highly modulated by all these complex functions of the hypothalamus.
Absolutely, yeah. It actually always confused me why we need so many regions for aggression. Because honestly, if you're thinking about if it just need to detect this cue and attack, that's like so simple.
Yeah.
The robot can definitely do it. You need a 1-year-old to do that calculation. So the fact that it has so many networks, it's a whole network and so much moderation going on, and so much flexibilities, plasticities. And probably more and more controls as the species gets higher levels in our case. I think, so that is an interesting question. And I think that's why it's hard to replicate in the robot, not because it's harder to make them attack, but just because how it's being all moderated. We still don't quite understand of that, how these contexts are being take into account.
Well, thank you very much, Dayu. It's been great having you here.
Thanks,
guys. It's been Neuroscientists Talk Shop.
All right. Awesome. Thank you. Thanks for having me.